Good evening, ladies and gentlemen. My name's Ingrid Winship. I'm the Professor of Clinical Genetics at the University of Melbourne and the Executive Director of Research at Melbourne Health, and in my latter role, a very proud sponsor of the Convergence Science Network, which brings you these fabulous events. Precision medicine is a relatively new topic, which has been brought about by all the changes in technology. So whether it's because of the Human Genome Project or epigenomics or any of the other omics or the recognition of all of these areas, it's been accentuated in the last year by President Barack Obama in his State of the Nation address, saying that he wanted to bring from the country who had done the Human Genome Project and got rid of polio, he wanted to bring the precision medicine into everybody's lifestyle. And in so doing, has announced a big initiative which will start with cancer in precision medicine. Now, genomics in the olden days, well, genetics started, and now genomics, the ability to look at all the genes at once, and it was enormously costly. But the cost of sequencing the genome is coming down, and the ability to get more and more information is growing exponentially, and it's now becoming a reality that we can actually do precision medicine. But what we need to do that is big data. So it comes as no surprise that tonight's talk is about the role of big data in precision medicine. Dr. Cecil Lynch is the Chief Medical Information Officer for Accenture in the United States. Now, there are a whole lot of interesting facts on that slide which I can't imagine anybody can read. But what I can tell you from my 36-hour experience of Cecil Lynch is that he's a gynae-oncologist with a profound understanding of the implications of technology on healthcare. So when he speaks to you about the use of big data, he doesn't do it purely from a theoretical framework, but he does it as somebody who's actually used big data and used technology in the management of patients in very dire need. I think it's no secret to anybody either that the Australian government has recently announced the Medical Research Future Fund, which is one of the most exciting things that's happened in recent times, which will put a lot more money into medical research. And while money doesn't create research, it certainly helps researchers to do good things. So tonight we're going to hear some fascinating stuff. Without further ado, may I invite Cecil Lynch up to the podium.
Well, thank you again for having me here. And I want to kind of start off. I'm going to give you just a little bit more background, just so that you know kind of how I am thinking about this as I approach it. I think Ingrid gave you a very real issue in that for 10 years of my career after my education, I did spend it taking out some of the nastiest cancers that exist: ovarian, cervical, uterine. I'm a gynecological oncologist by clinical training, and I think what that does to you as a human being, taking care of people with that level of suffering, is to hope that you are put out of a job as soon as possible. And quite a while back, I looked at kind of the direction that things were going. For me personally, I kind of started in an IT background, and then I was a chemistry undergraduate major, biochemistry, with a minor in mathematics and computer science, and so I kind of had a little bit of the computer science inkling. And then I went to UCLA and did the MD PhD, and my PhD work was in reproductive neuroendocrinology. So I had the bench side of it, and I did an NIH fellowship on looking at the effects of somatostatin and controlling the endocrine system, and then practiced, and then realized that I couldn't keep track of the information in my own practice. So started building EMR Elements, and then realized that as I could start to mine some of that data from my patients, I started seeing behaviors even on a small set of patients. And so when I negotiated with my wife for more children after my first 10 years in practice. She said, it's fine. I had 2, and she said, you know, it's fine, you stay home and change the diapers and do the bottle. Be a real parent. And so I took 3 years off from work and stayed home and changed diapers and did other things. And when I stopped that, I started saying, well, what can I do now? Should I go back to clinical practice? Should I look at other things? And I talked to my father-in-law, who's a nuclear physicist, and I talked to him about, well, you know, I've kind of always liked mathematics. Maybe I should do theoretical mathematics. Or he goes, it's a waste of time. Do something that's going to help people and something that you know. What about your background in computers and your background in medicine? Maybe you could do something else. I said, well, I don't really feel that good. So I went and did a master's in computer science so I could feel better about what I could do from a technology perspective. So when I think about this, I'm actually wearing every one of those hats. So what do I do on the bench side of investigating things? If I had to synthesize an estrogen molecule, or I had to, you know, chop off a number of rats' heads and take their brain and dissect that and find out what the hippocampus was doing, or if I had to do surgery on a patient and watch her family suffer and she suffer as I couldn't cure her ovarian cancer, to, you know, how do I put up a platform that's going to help thousands of people instead of the one person I can help in the operating room. So that's what I think about when I think about, you know, the changes in medicine, the way we're going and where we're going. So I want to now ask people with that background, so how many in the room are clinicians? Okay, so got quite a few. And how many people in the room are researchers? Okay, and how many in the room are computer science people or bioinformaticians? Great. So we've got a good breadth. I saw some people raising their hands multiple times. So I think those people really know what I'm talking about from how I'm approaching this. So I want to talk a little bit about kind of what does it mean to put together a platform and an approach to understanding all of the huge volumes of information that we're starting to get. And how do we apply that, and what is the best answer that we can get to drive that personalized care for an individual patient? And what does it take to put the pieces together? But if you think about precision medicine, what we're talking about, it's really identifying the specific therapies that are tailored not just to a person's genome, but to their preferences for care, for their individual lifestyles, for the environmental areas they find in themselves. And for other factors that we probably are not completely aware of, sentiment, you know, how they feel about things. And it's important to also realize that just finding that therapy is not enough, that it's also about giving the information to that patient so they can deal with that therapy and deal with what they found out about their own makeup. It is about providing that information in a way that it's ethically based, that you're not giving them a bunch of data about their genome when there's no treatment for what you're going to talk to them about. You know, if you're going to talk to them about a treatment option, talk to them about all treatment options, including the options of doing nothing, right? So those are definitely options for them. And don't forget the family, because when you start talking genetics, you're not talking about that individual, you're talking about their parents, you're talking about their close relatives, you're talking about the children that they will have eventually. So you can't forget the whole family. So what are some of the caveats that we think about of genomics-based precision medicine? So, you know, what does the sequence mean? And by that, I'm really talking about the things that I think I heard Ingrid actually talk about yesterday, I believe, that it was you, that we're talking about, you know, all the trash in the genome, the things that once were called trash, that we now know is not trash, you know, and there's going to be a lot more of that as we go forward and we understand more about the genome, is that some of the things that we have no idea what they do or think that they're inactive, we'll find that in constellation of other elements we find, other pathways we identify, the actions on specific proteins, that we're going to learn more as we go on. And I truly think that right now we're in the infancy of what we know and what we can predict. And I'm going to show you some information as we get to the end about why you can't throw away all the phenotypic information that we've had as predictors of disease, specifically in certain genomic areas, so that all the answer isn't in the genome. So as I think about, prepping for what we need to do to figure out the whole view of what's going to happen to that patient, and really think about it from a holistic point of view, not just the genome, not just the environment, not just the population ethics. We've got to really look at everything. We've got to understand the disease process, right? And I'll go through a use case a little later on about why it's important to understand all of that disease process. How do you get to an understanding of just the disease before you tackle the genome? Because when you tackle a specific disease, you may be missing the bigger picture. You're getting into the weeds, but you're not really, into the individual trees, but you're not seeing the forest around that sometimes. And then, of course, what's the pathophysiological nature of not only that disease, but the family of diseases that it's connected to? So targeting one disease may in fact increase your risk for another disease. How do we get enough information on those clinical conditions and the comorbidities, and then the treatment confounders? Certainly if I provide that daily aspirin a day, right? So it's a good example of this. So we know that taking a daily baby aspirin can prevent your heart attack, right? Theoretically. It also has a protective effect on colon cancer, right? Lowers the risk of colon cancer. But if you take a baby aspirin a day, you increase your risk of a gastrointestinal bleed that might be deadly by 7-fold, right? So it isn't without its own risk, the therapies that we do, that we're going to go through. And so then we have to start looking at not just the genome, but the environmental factors and the population health that we go. And then we're going to talk a little bit at the end of this about what's the information technology approach to that, both from a data mining perspective, about what we can learn from big data and large-scale phenotypic information, and then how do we move ahead to bring the phenotypic data together as we get more and more robust electronic information on the patients with the EMRs, and how do we then take that and put it together with those 1 million genomes that Barack Obama is going to get for us and the whole sequences, because there's going to be a lot of effort there on connecting that information. And I will say there's already quite a bit of information, and I'll present some of that, about looking at the predictive analytics of what the genome is telling us about what's going to happen to a person later on in life, and what the population health or the phenotypic view of that is. And I would like you to keep in mind that we are very nascent. So what we can predict right now from a genotypic expression, in some cases I'm going to show you, is no better and sometimes not even as good as what we get out of just looking at the phenotypic expression. We get no value from doing the genotyping. So that's hearsay probably for some people that are looking just at it from a genetics. And I only say that because, and I do not say that does not mean that there isn't value. It means that we haven't found all the value yet to where we're going to go. And one day we will. And we've had 50 years, 75 years of looking at phenotypic expressions to understand disease with very smart clinicians sitting and investigating that and thinking about what they see. We don't have that 50, 75 years of looking at the genome with our tools and saying, this is what all this means. We will get there over time. So how do you approach it? What's a framework that we can put together for personalized medicine, precision medicine, that encompasses all of those things? So conceptual map of the domain, so what are all the factors about a disease that we know? What are the therapies for that? What are the comorbidities? What are the genomics behind it? Both those that that are expressed in the blood and those that we have to get from sequencing, expressed in the blood from proteins that we know are related to that definition. Then we have to think about, if we're going to study this, we have to be very careful about deciding how do we constrain the world of all those things to get a problem that's specific enough to get an answer that's actionable, but not so specific that we ignore some of the impact that that specific therapy might have. And then we have to realize that at the end of the day, it is going to be a mixture of these. All of the phenotypic data that you have plus the genomic data, and there has to be a bridging of that. And it isn't going to come from putting together, 10,000 patients' information. It's going to come from putting together, 1 million, 5 million, 60 million patients. And when you start doing that, that data doesn't exist in one system. It exists across multiple systems. It's across multiple large hospital systems, all using different EMRs. It's across different metadata binding for even naming variants. It's across understanding the bugs that infect us and the naming convention for those. We have an entire international organization that judges what the names are for the next bacterium, right, or the next virus, because so many people come up with different names for these things. And so the terminology that's behind this and using careful, sound practices to have descriptions of these things both at a metadata level and at a datum item level that allows us to then aggregate that information is very important, because that's how your analysis is going to have meaning. You have to have a way to bridge all of this information together. So you have to have a data model that can handle all of the metadata about your sequence, but also all of the data about your clinical information, and binding that together where you can reach down and get to that extra information. And there's a lot of work that we don't have time to go into today that's being done within the standards world that's looking at that. How do we take these fast healthcare interoperability resources from the IT guys, these resources that will be able to move data around easily from an EMR, and how do we take that and then present genomic information in those resources, but also link out to something where we can get to the actual genome for this data and bind that all together into a single information model. And there is work being done to do that right now. Then we have to define the analytic questions that we want to answer, and there are going to be many of those, right? There are going to be many of them, and all you have to do is look at any mapping of any protein expressions for any cancer, and you'll see how many ways there are to get to specific elements of defects, overexpressions, underexpressions. And those pathways are very complex, and they're going to get more complex as we learn more. And then there's the analytic approach. What are the right algorithms to apply? Do you have enough data for it to be statistically significant? Do you need to add more data? Can you do surrogate trials for these things to improve the power of your analytics? And then how do you visualize that data? And not only how do you visualize the data as a researcher that has a deep understanding of this, but how do you present it to a layperson? How do you present it to somebody so that they have the understanding of what's wrong with them, so that they can talk to other family members about it, so they can talk to their doctors that don't have that genomic background about it, especially when they move somewhere else, or they get shipped overseas for a job, or they're in the military and they go somewhere else. How do they talk about that? So you have to think about something that distills it down to the individual's level of literacy and ability to comprehend the information that you've given them. So that's the kind of the key principles for the framework that you have to think about from precision medicine. So let's take a look at a use case. And this is something I've been studying for a while. I have access to a data set that the primary consumers of this is big pharma, because they're way ahead on a lot of this stuff, right? They're doing precision medicine now. They're trying to build targeted therapies based on the genetics of an individual. And one thing that they have found is they can't do it just with that. So they are the biggest consumers of big data from a clinical perspective. And so I have a repository of 60 million longitudinal health records that on average go back 7 years for an individual. It's EMR data, it's very rich. Sometimes it's back as long as 12 years. for some of the patients from systems that have had EMRs for a longer period of time. But the data is all standardized to standard terminologies. I can query it. It's all in a big data environment, so I get answers in literally a few seconds that I can do a query and execute it across those 60 million patients. So it's a candy store for a clinician researcher on these things, because I can ask all kinds of questions that I've had in my practice over time that I didn't have enough power in my data set to answer. And you can find many, many things that are not in the literature. So I'm going to talk to you tonight a little bit about something that isn't in the literature. So you won't read about this. You won't go to PubMed and find something on this yet. So what I'm going to focus on is Alzheimer's. And I'll tell you a little bit about how I got going on this. I have a colleague who runs the U.S. National Library of Medicine. His name's Clem McDonald, and he does the research for their data set, the NLM. And I was at a standards meeting with Clem, and we were at the social hour on Wednesday night, and Clem walks up to me and he says, "So Cecil," he goes, "I have a question for you. You're getting older." I said, "Well, thank you, Clem. That's very kind of you." And he goes, No. Yeah, he goes, but you think about your heart disease and how you're going to overcome it." I said, "Well, I don't have any heart disease." And he goes, "Well, no, but preventing that. Have you thought about taking a statin? Because, of course, statins have an anti-inflammatory effect on the coronary arteries. So even if you don't have hyperlipidemia, you can take a statin that will help to decrease the inflammation in your heart and likely prevent a heart attack. There's good evidence for that it lowers the risk of myocardial infarction." So I said, "Gee, Clem, I don't know, so to take a drug that, affects the lipids." I have a family member who was on a statin, totally anecdotally, who, comes from a family where everybody lives till they're 100 or older, and they die, sending dirty jokes on their Mac, from their bed where they're trying to decide whether they want to do the dialysis at 101 or not. So these are people that are with it, right, till the end. And and now I have this family member who's in her mid-70s and in rapidly progressive Alzheimer's disease. And the only difference is, she had a heart attack at 45. She was a smoker, and then she had some hyperlipidemia, and they put her on statins. Right? So she's been on statins for a long time, and she's had all your major side effects from it, the peripheral neuropathies, the other things. So I'm thinking, well, if you're on a drug that can give you all these neuropathies peripherally, what is it doing centrally? And so I kind of want to dig into my data set and see what the effect of statins are on diseases that have demyelination problems. I want to see what the effect is there before I make a decision on that. So I started digging into our data set. And if you think about the approaches to Alzheimer's, there has been a ton of work in Alzheimer's on the genetic basis, right? Because we have a great set of patients to do research on. Alzheimer's has a couple of different forms. So this is just a view of, really all the things that we know about the pathology of Alzheimer's up to that time. This was published in Cell, and it was really trying to break down all of the issues of the cell functions of what was happening in an Alzheimer's patient that changed things. And then, we started learning a lot more about the genomics. And why did we learn about that? Because we have those two forms. We have the autosomal dominant form of Alzheimer's disease that has an average age of onset of Alzheimer's in your early 40s. So, and even as early as your 20s or 30s, the first signs of starting to lose cognitive function. It's a devastating disease. And it is autosomal dominantly inherited. And we know what the gene mutations are for it. And so it gives us a target for drug development against those mutations. We also have the sporadic form of Alzheimer's, which is the typical thing we think about. The person gets into their 60s, 70s, and starts to have cognitive decline earlier than they should really have it, and then progresses quickly, which is associated with the hippocampal degradation in the brain that you can look at on MRI or CT or PET scanning and really see the physical changes that happen with that disease. And that has a different genomic factor. So it lets us kind of now target both of those types and learn from that. So what do we know about the successes and failures so far of targeting those from the pharmaceutical industry? Even with good markers and a fairly well elucidated pattern, no benefit. No drugs in clinical trial have had any benefit on slowing the progression of Alzheimer's disease in those patients who have had cases of certainly the autosomal dominant. The trials showed no benefit in slowing or preventing the disease in a group that you would hope that you could measure that well, because you know that whole group is going to get it, so you can start the therapy as early as possible in their history before they have signs, because you know they're going to get it. And no effect in tackling APOE4 yet in those drug trials. So we're not at a point yet where we can claim any successes at targeting Alzheimer's disease from a genomic perspective. So you have to ask, is the genome the entire answer? Is it something that we can really hang our hat on for all diseases? We've had great successes, I think, in cancer so far. We've had really good guided, targeted therapy where we can identify the defects. Now, we heard an interesting one yesterday that still opens up that whole baffling question, right? You're giving a drug where the genes don't match at all what you thought you were going to give that drug for. You expected that this person that you're giving a drug, that really isn't used to treat that kind of cancer, that has a miraculous remission for that cancer. You're sure that you had, that you targeted that gene that was responsible for that cancer, and then when they did the sequencing, that defect wasn't there. So it's something else. That was a remarkable talk yesterday by Dr. Shine. But it just tells you what we don't know so far about this. And there's another friend of mine who's at CMS, the Centers for Medicaid Services, Medicare Services in the United States. And they have a huge repository. It's mostly claims data, but it's claims across both the care you get, so it has all your diagnoses, and also the medications that they paid for. So everything that they paid for, they have a record of that. And they have close to 45 million longitudinal records going back lots of years on that, that they can mine that data. And one of the things that they found that were very interesting that Dr. Soros looked at, he said, well, gee, I'm kind of curious about what we're going to learn when Barack starts to get this genomic data, so I want to get a head start on that. I want to look at a cohort of identical twins, which should have the same genomic information, and I want to look at a paired cohort of non-identical twins, so I can look at environmental effect, and then I want to look at another cohort to compare that with. And his findings were very interesting. And what he showed is that they actually could only find one linkage that was truly outstanding based on the genomics in those patients when they followed the claims through. Does anybody want to guess what that might be? In twins. Atrial fibrillation, a highly genetic disease, and one that when one twin got it, the other identical twin also had atrial fibrillation. So it was pretty right on. But for most things, very little lift, right? So what they ended up on their analysis said, well, at best, in our twin study, we see that their comorbidities, the diseases that they have in common, about 30 to 40% of that has a genetic explanation in our twin study. So it was interesting. And that data was published in Population Health Management this year. So they're continuing to look at that and expanding that study. And I'm back there on Thursday next week, so I'll have more information about where we're at on that. So what about a phenotypic approach for Alzheimer's disease, and what do we see? So I'm going to step back a little bit, and I'm going to go out and look at my comorbidities, and I'm going to look at this high-level abstracted map about what makes up Alzheimer's disease. What can we look at from a genetic basis? What do we find of signs and symptoms for the patient? What are the drugs that are being used to treat it? You know, how do I get all that so that now when I start to do the data mining, I can one by one walk through all of these variables and pair it up and take a look at this so I can see phenotypically, is there something there? And so there was something interesting. So I had a reason to do this, because now my colleague is saying, all right, take this statin. I'm saying, okay, it gets back to the options. You know, what options do I have if I'm a hyperlipidemic? If you then come and tell me that maybe it's not the best thing to take a statin if you have Alzheimer's, if I happen to find that. Well, fortunately, there's a new drug coming in, still in stage 4 clinical trials, class 4 trials right now, almost ready to release, the PCSK9 class of drugs. People know about this drug? So it's an injectable, once a week, sub-Q, pretty easy to take relative to taking a pill every day, and it is remarkably more effective than a statin. So what the FDA has given preliminary approval for this drug is in cases of patients who are refractory to the statins, so it doesn't control their hyperlipidemia, patients who have familial hyperlipidemia where statins just do not control it if it's on that genetic basis. They're going to produce on it, and you just can't lower it with statins adequately. And then patients who absolutely completely fail a statin because of side effects, you know, bad neuropathies, other things, they can't tolerate it. So that's their use cases. So that's about roughly around 10% of the population that have high cholesterol levels. So what if, though, that drug doesn't have the same side effects on Alzheimer's disease? Maybe it has a different effect. Well, it has a different mechanism of action than statins do. Statins work on the enzyme HMG-CoA reductase. They're inhibitors of that enzyme. And so they stop, they have a pathway that looks at inhibition of the synthesis of it. Clearly they can't block it all because you're still making lots of it, right? And that's why some people fail the statins or they get marginal control. And it's why it doesn't block it all. PCSK9 drugs actually work at the receptor level, and they internalize the receptors and bury them back in and then consume those receptors so that you can't make it because you don't have receptors that can be expressed to allow you to make the cholesterol. Highly effective at doing it. Maybe too effective, some people think. And so when you think about PCSK9 drugs, there's been a bit of a concern that it might cause issues of things like Alzheimer's disease or other demyelinating diseases, multiple sclerosis perhaps, because it lowers your lipids too low, right? You get down to 60s and 70s on total cholesterol. So do you have enough to use that for myelinating the nerve fibers? So it's all a very interesting question. So what do you do when you're mining that, and what did I have access to? So I had a base. If I wanted to look at those people for sporadic Alzheimer's disease, I'm going to look at the group of patients in my data set that are over 65. I have 10,400,000 of those patients that I can look at. So now I can really start mining that. And in that group, I had over 1 million of them that have hyperlipidemia. So I have now a pretty good set, so I can start looking at that. And in that cohort, I also had almost 200,000 of them with Alzheimer's disease. So we're starting to get to some pretty big numbers to start really looking at things. And so I started mining that data. The one thing I would like to remind you of is that this is observational data. And observational data is not basic science research, right? So there may be nothing that comes out of this at the end of the day when it's goes through clinical trials and other things. So keep that in mind. It's not something to quickly run out and stop your statin if you're on it already and you have high cholesterol. But it's definitely something I'd be following the literature as it starts to come out. And this study is in the hands of Big Pharma now. I've made it available to all of the ones who are building out PCSK9 drugs in the protocol to really start taking a look at those patients longitudinally in their clinical trials and go back and look at these things. So everybody's going to be looking at that. So what did I find in that? So if you have, in that data set, if you're over 65, about 1.7% of that population has Alzheimer's disease, right? And if I put those people on, if those people have hyperlipidemia along with that, then you see that there's a pretty good rise in that risk of Alzheimer's disease in that set. Right? So hyperlipidemia, and if you look at the literature and you mine PubMed, you will find that association between hyperlipidemia and Alzheimer's disease. So that's known. Right? But look what happens when you add a statin to it. You bump that up to 3.9% of your patients have hyperlipidemia. So this group that I show here with the no hyperlipidemia, that are on a statin is my control group. And that's what I went back to before and talked about, that some doctors will start you on a statin not because you have hyperlipidemia, but to reduce your risk of heart disease. You have a family history of heart attack in your family, so they're going to use that anti-inflammatory effect of the statin. So we see in that group that are on it not because they have hyperlipidemia, that they still have a really high risk of Alzheimer's relative to your control group. But if they do have hyperlipidemia on that statin, it raises it even further. So, and also, if I have Alzheimer's disease and I don't have hyperlipidemia, then if I don't have hyperlipidemia and I'm not on a statin, then my risk of Alzheimer's disease is about 1.6%. So it's significantly lower. Now, what does that say? General insights, right? So if you're on a statin, you may be at higher risk for Alzheimer's disease. If you have hyperlipidemia, you're at higher risk for Alzheimer's disease. If you don't take a statin and you don't have hyperlipidemia, you're probably at lower risk. So that's what we can say so far. Now, there were interesting things because I said, well, what is it? Maybe it's, how do I explain that? I don't have a good explanation for that. Maybe it's something else. What happens, what's the end stage of hyperlipidemia? What's coronary artery disease? And what's the end stage of coronary artery disease? You have a heart attack, right? And if I looked at those patients in my group, and I looked at those that start to get, that have heart attacks, well, you can see pretty good. If you have hyperlipidemia, you're at a higher risk for that. But interesting, if you have hyperlipidemia and you're on a statin, you have a higher risk of heart attack than if you have hyperlipidemia and you're not treated. So that's interesting, right? So is there really a protective effect of the statin on the coronary artery in the case of hyperlipidemia? I don't know, but it's worth looking at, right? So you want to look more into that. So this is also one of the things that the clinical trial folks at these pharma companies are now starting to look at. But look what happens if you treat the patients who have hyperlipidemia and have coronary artery disease versus don't get to them soon enough and they have a heart attack. And mind you, I excluded in this group another type of dementia called vascular dementia, right? So it's been excluded. So you have in our data set, we can do that. We have a rich classification of all the dementias, and so we know those that have a diagnosis of vascular dementia versus those who have Alzheimer's disease. But look what happens. If you get a bypass or a stent and you open up and you prevent that coronary artery plaque buildup, it's having an effect to lower the risk of Alzheimer's disease. Whether you get a bypass, whether you get surgery or a stent, right? Whether you get percutaneous surgery or whether you actually open the chest, you get an effect. If you have an MI before you do that, and that happens before you get to the OR and you get that stent, and you survive it, there's a high risk of Alzheimer's disease. So why is that? Is it acute phase reactants that are released with the heart attack? Is it, a period of hypoxia that occurs from loss of blood flow? Is it the fact that those people that have a heart attack before they have had a chance to have an intervention, or in other words, they don't have signs or symptoms earlier and they have the heart attack, maybe they have a greater occlusion within the brain as well in the circulation, so it leads to some of the effects of the Alzheimer's. I don't have an answer for you there, but we're looking further into that. So you may ask, well, maybe those people that are in those different groups, they have different lipid levels, or they have hypertension, or they have something else. No. There's no difference, really. No difference. These patients have pretty much the same systolic, pretty much the same diastolic, pretty much the same cholesterol levels. It wasn't any difference in those cohorts that are treated, not treated groups. So that's not the answer. And is it just for that particular disease? Is it just for Alzheimer's? Or can you think pathophysiologically? What are other demyelinating diseases that might be affected? Well, so I said, well, let's look at other demyelinating diseases and look at other neurological diseases that are not demyelinating in nature and see what we see what the effect is. Well, look what happens. If you look at multiple sclerosis and you look at these findings, you'll see that, gee, if you look at seniors on the statins or with hypercholesterolemia, you start to see more of the multiple sclerosis than you do when they're not on a statin or they don't have the hyperlipidemia. So there's something there in that myelinating disease as well. And if you take a look at other diseases that are not demyelinating, like Guillain-Barré or like ALS, there is no effect, right? These are severe diseases that have neuropathogenic findings with them. Got another neuropathic disease, but they don't have the demyelination as part of that process. You don't see an effect. Parkinson's, again, you see an effect. It's a demyelination disease. So there's something there about the fact of, is the etiology is a demyelination process, whether it's, the case we see in Alzheimer's disease, which is not pure demyelination, it's abnormal myelination. But if you see abnormal myelination or demyelination, there's some difference there for that, and there's an effect if you're on a statin. So certainly it's a research question that begs to be answered for this, because huge numbers of people, as you've seen in this cohort, 1 million of them have that. And that is 2.5 times all the new cancer cases in the United States in a year. Right? So it's a big number, it's a big disease, and it's the #1 killer. Right? So as important as what our genomic findings are in cancer, wouldn't nearly have the effect if we have a genomic approach to identifying these patients and ameliorating that. Huge issue. So these are the top comorbidities that you see in Alzheimer's disease with more than 1 in 5. So they have hypertension, they have arthropathies, they have hyperlipidemia, they have renal impairment, type 2 diabetes. So you've got to look at these now too. So we've got to do the due diligence to dig through the data sets and look at each one of these comorbidities and try and factor out whether it is. But because we have all that data, we can do it. Now then once we find it, then we can go through and we can look at some of these diseases that in fact do have a way to look at them from a genomic level as well. And so when we get the 1 million genomes, we can now start to look at that in cooperation with our phenotypic data and start putting these things together. All right, so I think you get the so what's. I'm going to skip this slide because the so what is, well, from a pharma perspective, if you're a CEO of Pfizer or Genentech, you're thinking, oh, if statins are causing this and all my statins are going off patent, great. I'll say, we didn't know that before. Now we've got that. We've got this PCSK9 drug that works. So instead of 10% of the population and a $2.5 billion drug, maybe we have a $12 billion drug because we're going to use it across everybody. So for those guys, they're probably pretty happy if they can find something like this that they don't find with that. For the everyday person, it means that you don't have to deal with neuropathies. You don't have to deal with the side effects of statins with a drug that might be better for that. So there's a big thing. So now I want to kind of show you a little something about what I said before about what's the lift of genome. So Soros kind of showed that in his twin study. So about 2 weeks ago, I was at the Individualizing Medicine conference at Mayo. And Dr. Ruth Luce, who's at Mount Sinai, had a presentation. And her whole presentation was about looking at a number of common diseases and doing predictive analytics on a phenotypic expression versus a genomic marker expression to be able to determine whether or not these people, you had the ability to predict it based on one of those profiles better than the other. So these are her slides for the next few. I want to give her credit for that. So these are the things she looked at. She looked at obesity, she looked at coronary artery disease, she looked at type 2 diabetes, and she looked at the genomic markers for these in patients who had the genomic sequencing or the biomarkers for them, exome sequencing in those cases, and then those that you just simply went off their phenotype. And here's what she found. If you look, and the things to really look down are right here. So if you look at the predictive value of traditional phenotypic risk factors, here's your ability to predict the outcome. If you look at traditional risk factors plus genetic risk score, maybe you get a little better, but not much. It doesn't shift things much, right? If you, and that's in diabetes, right? Look at coronary heart disease, where they're pretty good markers, right? And here she looks at it again and she says, well, what if I just look at traditional risk factors for coronary artery disease? And now I look at genetic risk factors plus my traditional. I get no lift at all from the genomic side of this. It doesn't improve my ability to predict, even if they're carrying all these markers. All I had to do is really just look at their family history, their lipid levels, and other things. And I got just as good as I would get from that. Same thing is true of myocardial infarction. Didn't make much of a difference, right? I couldn't get any more out of, I got no benefit from doing the genomic evaluation. So I want to end that with saying what I said up front. We've had 75 years to understand the phenotype. We haven't had very long at all to understand the genotype. And I think this will change as we get smarter on what we know about the genotypic expression. I think this is a point in time we're going to look at this evaluation and we're going to look at it in 5 years from now and we're going to say, well, they weren't looking at the right pieces of that. So now I'm going to talk just very briefly about the technology stack. This is the technology stack that I use for the data mining. This is where those 60 million people are. And it's pretty good for that. It's all based on Hadoop, a big data file system that was contributed to us by Google. It's what Google and Netflix and Facebook and everybody who mines big data out in the world are all running on these big data platforms. All of them are based on Apache Hadoop, to some extent. We use a NoSQL database because it's not very, it's great to get every bit of your data into Hadoop, store all of your data. You can ingest it pretty quickly. You can store anything, but once you try and get it out, it's pretty slow to mine that. So you need what we call a NoSQL database to do that. Very fast query engines to get there. And of course, all the analytic tool sets that are up there on top of that. Spark is an in-memory analytic tool. Very fast to do any kind of an analytic, genomic sequence analysis, because you're not running on data in a database, you're running on data in memory. So it's very quick to do these kinds of things. And then visualization tools that we put on top of that. This has a HP Vertica, which gives you a relational database model so you can execute standard SQL queries against it. You don't have to know MapReduce or write, the Java code that normally you would write to get to that stack. And then SAS or Tableau on top of that. So that's what we do with that. Would this work purely for genomic data and aggregating that? No. So here's what you look at for a true precision medicine platform. So what are the important pieces there, right? So you've got to have, a ton of this stuff that's out in our medical records is in text, right? So you gotta parse that, you gotta understand it, you gotta write it to some model that you can do more robust analysis. If I want to, though, analyze my genomic data, so you all know about the Global Genomic Consortium. Melbourne is part of that, right? So they publish a ton of useful tools that are meant to process this data. And one of the ones that they publish is how you do the reads, writes, variance analysis, sequence analysis on data as you're streaming in to a platform like this. And it's done with something called Apache Avro. And Avro works in another Apache tool called Flume, which is a streaming ingestion. So if you've got Illumina that's producing your sequence data and you're piping it in through Flume to go into this rest of Hadoop where you can then do further analysis, you can do a lot of your analysis right on the fly through things that are produced by the global consortium. And those are meant to run right in this big data stack. So, and you can download them for free. So this is the kind of platform that you need to do that. It gives you all the scalability to store any amount of information that you need inexpensively because it's open source. It doesn't cost anything, but it's open source that's totally supported by Google, Netflix, Yahoo, everybody else. So it's not your mom and pop's open source. It's your production level that every company that's running big data runs on. And the tool sets that give you the analytics that you have specific for genomic data. So this is what you need to also port in all that relational data that you get out of your EMRs and combine it in that Hadoop stack so you can do your genomics mixed with your data that is coming in from your EMR systems. So I'm going to stop there. Again, thank you very much for the invitation.
Well, thank you. So, that was an absolute tour de force of informatics, and I'm going to go home and see if I can remember if I'm on statins or not, because that data is quite alarming, and if indeed it is correct and it needs to be put to clinical trials, it will have a profound impact on the health of so many people around the world. Firstly, to thank the sponsors, of whom there are many. Without sponsorship, nothing happens. The network, Convergence Science Network, is having its 7th birthday in October. And so far, there have been more than 21,000 people attending events for the network. Lots of speakers, lots of people in the audience, lots of people have made this network the exciting thing that it is. So if you would all join me now in thanking Cecil for a marvelous talk and a lot of thought-provoking ideas to take away.
Thank you.